Hello everyone, my name is Yihong, and today I'm gonna introduce you the microplastic pollution in deep sea sediments and organisms of the Western Pacific Ocean. And speak of the microplastic, we have to know the plastic amount of plastic into the ocean. In previous studies, we have known that the annual plastic production in the world is about 350 million tons per year in 2018, and 10% of them will eventually enter the ocean. And those plastics, their sizes are ranging from meters to nanometers. But in this study, we only choose those who are under 5 millimeters as microplastics. So, how does the microplastic affect the organisms? First, it will increase the energy expenditure, means you have to use more energy to eat the microplastic and excrete them. And it will cause physical blockage, means it will stuck in your body, and it causes false satiety. Satiety means if you eat something and you feel full, the feeling of full is the satiety. And in microplastic, there are mostly harmful additives in the microplastic, and it will release in your body like pesticides. This is the substance we use there in the plastics production. It will make the plastic more tender or soft, and it's harmful. And the microplastics will absorb the persistent organic pollutants, we call it Pcbs, and heavy metals, and both of them are toxic. And I have to introduce another substance called Pcbs, and it has a strong toxic. It will cause cancer and neurotoxicity, and it's a kind of persistent organic pollutants. And persistent means it will stay in the environment for a long time. And this Pcbs is widely used before since 1930s, and is banned for use since 2001 in the world. It's mostly used in engineering products. So our research purpose is to understand the distribution and abundance of the microplastics and Pcbs in the Western Pacific deep sea area, and to more comprehensively understand the correlation, means the relationship between the microplastic and Pcbs, and to investigate the microplastic contamination in the deep sea organisms. So this is our sampling size. As you can see, it is a little bit small. Here's Japan, Korea, China, and Taiwan here. We'll talk about this there. So how do we get our sediment samples? We use the box core to get the sediment, and we will retain 0 to 5 centimeters of surface sediments in the glass bottle with aluminum foil for the studies, and we store them in 4 degrees centimeters. And for biological samples, we collect them with the ROVs. You're familiar with that, equipped with the mechanical arm here, and the suction hose, and it's the tube here to suck the biological samples this is to keep the samples to be complete. And these four pictures are the kinds of creatures we collect, and we classify them to orders because we don't really need to know what species they are. We only need to know if they're microplastics. So for the sediments, we separate them into 2 portions for analyze, but we will only talk about the microplastic analyze here because this is too long. So how do we separate the microplastics from the sediments? The first, we add potassium hydroxide to degrade the organic substance, and add the saturated sodium chloride solution and stir them. Then we centrifuge them, and we get this. This is the floating microplastic and the solution here. And the sediment we don't need to use here. And we collect the supernatant. The supernatant means the liquid up here, and repeat the 3 steps for 3 times. And the fourth step, we use filter to filter the supernatant with process at micrometers, and then dry the filter paper for further analysis. And for the organism samples, it's very likely we dry the sample and degrade the organic substances, and we filter the whole solution because there's no sediments here, and then dry the filter paper. After we get those microplastics, we first using the microscope to characterize them with their size, their shape, and their color. And then we transfer them to analyze using the micro-FTIR. It uses infrared. Infrared is a kind of invisible light to get its spectrum. And compared the spectrum, we can get the polymer types of the plastic. And after that, we get this result. As you can see, we have the average of 240 microplastics per kilogram surface sediment. And here is from 21 degrees to 24 degrees north latitude. Has the most abundance of microplastics, and our average is actually higher than other deep sea area like Arctic area. We have 240 here, and they have only 44 here. And our data is more likely to coast area in Malaysia and Bay Area in Thailand, and that's because our sampling sites here are very close to the Western garbage patch. So our main source of the microplastic might come from here. The concentration of the plastic on the seafloor is over 3,300 items per square kilometer. So the microplastic, their colors are mostly blue and fiber in shape. And their sizes are mostly under 500 micrometers. And I have mentioned that sizes under 100 micrometers is not counted because we can not characterize them with their shape, and their polymer types are mostly PE and PET. So the concentration of Pcbs, let's compare it with the abundance of microplastics. That you can see here, there are much higher than other area. And in statistics, we found that there are high correlation between the Pcbs and microplastics. So we can check the first and second purpose, and we know the distribution and the abundance of the microplastic and Pcbs. And we also know there's indeed a correlation between microplastic and Pcbs. In biological samples, the microplastic are all fiber, and their size are ranging from 100 to about 700 micrometers, and there are 9 to 3 items per sample, and their colors are mostly red, black, and blue. So we know that the microplastic indeed enters the inner of the deep sea organisms. So we can check the third. We know there are indeed microplastic contamination in the deep sea organisms. So how we have a problem that we found that there's high probability of false microplastic. False means about 76% of them are fake. How do we improve it is to dye the sample with fluorescent dye. This kind of dye is specially bind to the plastic. So we in microscope we can easily identify the microplastic for the first. And thank you for listening.
Because you talk about the Pacific garbage patches in the slide, and I want to ask you to explain how are the patches formed.
How are what?
How are the garbage patches formed?
It's from.
Repeat her question.
You want to know how the patches form. That's because the circulation of the sea current will make or form a gyre in here and here, make the plastic will dump into the ocean, floating on these, mostly on these sides.
I would like to add the basis of choosing the sea area where the research proceeds.
Repeat your question again.
The basis of the sea area where the research proceeds.
The basis of the sea area
where the research proceeds. Yeah.
So you want to know how do we choose the site to send for research? Yes. Actually, according to the, we do this because we want the data here about the microplastics or the in the deep sea organism research here is pretty less. So we want to know more about this.
What is POPS?
POPS.
And can you explain?
Okay, POPS is a kind of organic pollutants, and this is mostly toxic, and the P persistent here means it can stay in the environment for a pretty long time, and this is the meaning of POPS.
Can you show me the image you show for situation image?
The image for what?
Pacific.
Pacific.
Yeah, this one. And you talk about you think you find that if the data is close to the Thailand Bay in Malaysia. Yeah. And but why the part is close to Malaysia and Thailand, but it's lower than the higher part.
You want to know why is the data close to the Malaysia and Thailand Bay and coastal area?
The higher part of the image is close to the data in Malaysia and Thailand, but the lower part of the image is closer to Thailand and Malaysia, but they are lower in the data.
In our data, the average of is the comparison is based on the average of our all data, all sampling sites. So.
Okay, you are not answering each other, so okay, let's move on. Sorry, because you are not answering his question.
Can you talk again biological
collection machine is cool. Oh, okay.
Repeat.
You want me to introduce the biological sample machine again. We use the remotely operated vehicles. It's the ROVs. We are familiar with that. It's a kind of machine we can work under the seawater, and we equip them with mechanical arms here, and bind it with a suction hose. We can suck the biological samples with this tube, and in this way we can do our best to keep our biological samples to be complete.
Okay, so I think he gave a very nice talk. So the first thing you notice is that he spoke very with a proper rate. So he was not really talking fast, and he talked very calmly, smoothly. So it's easy to follow his talk. And another thing you notice is that he actually used a lot of icons, as I mentioned in the first lecture. There's abundant resource in PowerPoint you can use to enrich your presentation. For example, here he talked about this blockage, physical blockage. Then he used this icon of a stomach. You can easily find it in case you missed the lecture. You can easily find icons. You can easily find icon here. Where is it? No, not this. Yeah, somewhere. Insert. I know, this is not the same as my PowerPoint. But anyway, you can find icon here to enrich. And as I always say, I want to emphasize 1,000 times, one image worth 1,000 words. So that explains this nicely. Okay? And then he also had this table, which is a very simple table, and I don't remember if this table was in the paper, but anyway, if there was in the paper, he made the table himself, and then he simplified the information, so it's easy to see the key information he wants to convey in his talk. And then you also might have the feeling, as I had, that his discussion was very interesting. That was because he used a lot of supporting images. This image was not in the paper at all, but you can, nowadays, you have Googles to help you. So you can find a lot of images that to help your presentation. You can just assume that when he was doing that, he just Google Western garbage patch, and then you can find this image on Google Image. So that's what you should do to enrich your discussion, because very often discussion is the most difficult part to interest the audience. And then also he always discuss right after the data. So instead of like presenting all data all together and then discuss all together, he present data and discuss data. So it's easier for you to link the data to the point he want to discuss. And then he frequently, also the same in Jaden's talk, you frequently use this technique of checking the boxes. But I would say that probably you don't need to do that often. You can probably do it once, because I have a feeling that you spend too much on this kind of slides. You can do it once here, after in your purpose slides, and then do it once again. Where is it? Thank you, before thank you. Do it once again, and then when you do that, please don't say research purpose. You should say conclusions, okay? And then you should not just check, because that it seems like there's no further information. It's better that you can write something more, a short sentence to tell us what is the extra information this study found during this research, not just checking the boxes. And then what Yi-Hong can further improve was, I had a feeling that he spent too much introducing this yeah, I think that's too much. It's nicely done, but I think this can be condensed. And what can be added in his next talk is actually in this presentation, that was just one pie chart where he talked about how the kinds of plastics found out in these samples, and how did the scientists know that, which plastic is what. This is actually based on the instrument that you already introduced in the method, but you did not introduce the raw data. So if next time you have condensed your method and you have removed that many purpose slides, then you can consider adding this. And you don't need to show all of them, you probably just show 2 examples. Yeah, because that's a very mature method to determine the type of plastic. Okay, thank you, Yihong.